So hello and welcome everyone to this live webinar. Thank you for tuning in. My name is Benedikt Weiss, and I have the honor to be the moderator of today's session, where we will demonstrate how the assessment of brain morphometry and connectome measures enables the development of more precise imaging biomarkers in neurodegenerative disease clinical studies. So the webinar structure is as follows. First, we will have a 30 minutes presentation and live demo of our use case provided by my colleague. And yeah, after that, we will have a 15 minutes Q&A session. So you're all muted, but you can put your questions into the chat during the presentation. And yeah, I will collect all your questions and we will answer them afterwards in the Q&A part. Before we start, I would also like to give you a short introduction on what biomarkers is doing. So in a nutshell, we provide semantic data integration and knowledge management solutions for the life sciences. And yeah, with this expertise, we run and develop clinical information systems, R&D infrastructures for companies, clinics, and academic institutions. Biomarkers is a spin-off of the Max Planck Institute with more than 20 years of experience. And yeah, our technology has been validated through multi-year contracts and thousands of users worldwide. Most of our employees are PhD level life science and software experts. And yeah, we are headquartered in Planegg, that's near Munich in Germany. And we are also certified according to ISO 9001 and 27001. And now I'd like to welcome my colleague, our brain imaging expert, Dr. Markus Butz-Ostendorf. Markus will now show you how to make the most advanced neuroimaging processing pipelines available for Alzheimer's and in general neurodegeneration clinical trials. Okay, Markus, the stage is yours.
Thank you very much, Benedikt, for your kind introduction. Indeed, today I would like to show you how to increase the prediction quality of your neurodegeneration and Alzheimer's disease clinical trials with the help of NICARA, our software product. So, to arrive at optimal prediction quality for aging studies and also neurodegeneration studies is quite a challenge. So typically cognitive testing is being used, but also if you have MRI available, Boxerbased morphometry is being used. The problem with cognitive testing is that it's time consuming and expensive. And the problem with Boxerbased morphometry is, although it's widely used, it's only of moderate prediction quality. What we suggest here is to combine and extend Boxerbased morphometry by latest neuroimaging technologies. And these are, in this case, surface-based morphometry and also surface-based DTI tractography. The reason why this is not routinely done in clinical trials so far is because this is very demanding technology, requires expert knowledge and also sophisticated hardware. And here's the solution I'm presenting to you today, namely NICARA. This is our software product, and it stands for Neuroimaging-Based Connectome Assessments in Research and Application. The novelty of NICARA is that it runs processing pipelines for Boxerbased morphometry, but also surface-based morphometry, as well as surface-based diffusion tractography imaging fully automatically. You get tools for visualization and exploration of extracted features. You can do cross-sectional as well as longitudinal multimodal comparisons, and finally also statistics within one and the same tool. Let me show you the typical NICARA workflow to you in this introduction here. So let's assume you have nifty data acquired from the scanner, from conventional MRI sequences, but also diffusion tensor imaging sequences. You have them available on your hard drive or locally. You can upload them to the NICARA server. So NICARA comes as a client server solution that can run in-house or as a cloud solution. Now the data is being uploaded to the server. And once this is finished, you can start the processing pipelines. Processing pipelines are depicted here. So here's an example for our morphometry pipeline. So we use latest tools and most precise tools for image registration and segmentation like FreeServer and Cat12 toolbox, do defacing, etc., and then the Atlas registration. So the tools are validated by hundreds of peer-to-peer reviewed journals, and especially for our DTI tractography pipeline that I show you next, this was even suggested as the preferred neuroimaging pipeline for DTI tractography being used in neuroimaging and neurodegeneration studies. Now, the good thing here, or the particular novelty of our approach in NICARA, is that morphometry pipelines, or surface-based morphometry pipelines, and DTI tractography pipelines use the same pre-processing tools. So that's why morphometry and DTI tractography are fully mapped to each other and can be directly compared. Once the pipeline is executed and finished, you will be notified via email, and then you can explore the results. So the first thing you will typically do is you will check the quality control of each processed workflow. So here the image segmentation shows the superior quality of the tools we use. So we have a perfect registration of these different brain areas. Now, then you get a report of, for example, of the image quality. That's very important because you need a good image quality for the register, or the assessments of the surfaces and also the tractography. So easy to interpret. School ratings here tell you what your image quality is, and if it makes sense to really include these data sets in your clinical trial assessments. Now, you can directly explore the results of the processing pipelines against the background of the nifty raw image, the T1 weighted image. So NICARA computes all these regions of interest here indicated as transparent spheres that are used for the surface-based morphometry assessments, but also uses the same ROIs as starting points and endpoints for the tractography, the DTI-derived tractography, and computes the tracts here. So that's why these results are fully matched to each other, and this also leads to really a superior quality of the rendering of the surfaces as needed for the tractography. Now, since you have these values, these results available, you also get the quantitative results, the quantitative assessments of the different modalities. So for every region of interest here, every ROI in this column, you get the different surface-based morphometry measures. So in this case, cortical gyrification, cortical depth, area, gray matter volume, and cortical thickness. And that's the interesting thing here. You can immediately, for every ROI, combine DTI-based, so connectome-based measures. So for example, the degree tells you how many incoming connections every region of interest has, depending on the tracts that were measured. So this is a connectome measure that tells you something about the hardness of the individual ROIs. This is a measure that's beyond the surface-based morphometry mapping. This becomes possible because we use the same common brain atlas for all the measurements, all the regions of interest. Now, the results that you see here can also be visualized graphically. So in this case, we have a 3D lattice graph of some of the surface-based morphometry results. For example, gyrification is here color-coded of these circles. So every circle is one region of interest, and the color code gives you the folding, the gyrification index. Now you can combine this information with DTI cryptography, so connectome information. So every connection here, every line is one connection, and the color, the thickness of the line and the darkness of the line indicates the structural connectivity. So you can directly explore both types of measurements with each other. NICARA, in addition, provides multiple options to filter your results and explore the results. So, for example, you can use disease-relevant anatomical or functional subnetworks. You can define them by yourself and use proprietary ontologies to filter these regions of interest and their connections. So the benefit of using NICARA for you is that you get a more precise patient stratification using all these measurements. You can do a better monitoring of treatment outcome, well, you have a lot of expert knowledge captured by the tools or provided by the NICARA tool, and also provided by biomarker support. And if needed, we also provide you the hardware resources by using our cloud solution. I will demonstrate the power of NICARA applied to a voxel and surface-based morphometry study here in the context of Alzheimer's disease patients. And the research question that we try to address here is the following. So does a drug candidate slow down neurodegeneration in treated patients compared to matched placebo patients? This is a question very relevant to you doing clinical trials and the reason to buy NICARA. The thing here is that we're not able and not allowed to show you proprietary trial data that we have, but we can modify the research questions a little bit. So instead of looking for treated patients, we take Alzheimer's disease patients from the semi-public database ADNI and show how they change in morphometry compared to age-matched cognitively normal controls. So the method we applied is the following. We used 25 AD subjects against 25 cognitive normal subjects within a 1 year longitudinal assessment interval from the ADNI-3 study. So we applied Boxerbased morphometry of especially subcortical ROIs and ROIs of subfields, and also we applied ROI-based surface-based morphometry of the cortex. We then show individual t-test statistics between AD and CN group for the different measurements. All measurements are normalized for the 1 year longitudinal time interval, so we use the annual percent change as an indicator of change. Now I show this live to you using NICARA. So this is the landing page of NICARA. You have the different sections: connectomics, morphometrics, transcriptomics, everything you can do with NICARA. We now focus on morphometry for the beginning. And that's why we enter the morphometry browser. So before we start the actual research, we just check what's there. So we have availability tables for the different projects and studies. So for example, we can see here an availability table for the ADNI project. So that shows all the subjects here that are available in ADNI with the different studies and the different visits. So where we can select from results and data. So now let's go to the actual assessment. I will show you how you can use these different data sets from different studies and run statistics in NICARA. So assuming that we have processed data already, as I showed you in the PowerPoint presentation, I will now show you how to create a surface-based morphometry longitudinal statistics. So first of all, you can select from a study. So for example, here ADNI data, the different ADNI studies, you can select one or more. So here could also appear your data. So if you have studies processed in your system, proprietary data, for example, you can also choose this or can combine with ADNI data, for example, to compare your results with ADNI data. Then we use a filtering technique to select the cohorts, to create the cohorts on the fly in the system. So first of all, we create a patient group. So this goes very quick. So we just say here cohort, and then we type patient. You see in the table here in the column the cohort. So you apply this filtering. Now these are 25 subjects. We move on. So NICARA remembers your choice and define the control group. So for this, again, we select the cohort, type control. So we match the numbers here, and then we move on and select the regions of interest. So we have 360 regions of interest, or 180 per hemisphere, according to the Human Connectome Project Atlas. For the moment, I select all of them. I go to summary. I can review my choices, and then I could just type a name, example, test run webinar, and select the project, and I start the processing. Now this will take about 15 to 20 minutes because you have to compute all these attributes, 5 attributes for 360 rows with all the statistics. We don't want to wait for this. So I put this into the background, and I will show you results that I prepared for you already. So with this, I just go back to our landing page, and let me start with the Boxerbased morphometry to show you this. Here we have the Boxerbased morphometry, again the same subjects, and so you can see here the report. It's an univariate statistics, longitudinal Boxerbased morphometry, 25 Alzheimer's patients. These are the first group, and 25 subjects cognitively normal. They constitute the second group. There are 87 ROIs included in this comparison, and the first graphic shows you the different regions of interest for which we found significant differences in annual percent change. And in the right panel, you see the localization of these significant ROIs. So as expected, the hippocampus decreases stronger compared to matched control. So deflections of the bar to the left indicate a stronger decline or decay in volume measured by Boxerbased morphometry compared to healthy subjects. We can go down. Here's a a histogram, you see all the annual percent changes, irrespective of whether they are significant or not. And you see how these two groups differ from each other. So the green is again the patient group, and gray is the control group, the second group. Below you see a tabular listing, so there's quite a strong effect size here for both the left and the right hippocampus. If you do multiple testing correction, these are not really significant. This is just for the individual testings where we see these results. So that makes sense to look at surface-based morphometry and to see how we get even stronger outputs here with this small number of subjects. So if we go back and look at the surface-based morphometry analysis, then we click on this link. We can again review these choices. So again, this is a 25 patients against 25 controls. So it's an univariate statistics of the longitudinal SBM. And then we can go down. So here we have the different measures: gyrification, cortical depth, cortical thickness, and cortical area, and cortical volume. If you stick to that one for a moment, we have a high number of regions of interest where we see a deflection to the left. So with these 360 measurements, NICARA reports much more, or many more regions of interest which are significant. So in total, these are 108 regions of interest which are significantly stronger decaying in Alzheimer's patients as compared to matched controls. So, and you see this here in the distribution of these ROIs, what the difference is compared to Boxerbased morphometry. You can also see this here if you look at the histograms here. You can see that the groups left, green is again the patient group, gray is the control group. You see that they are much more different and separatable compared to the Boxerbased morphometry. So before I wrap up these results, I would like to give you an outlook what else you can do with NICARA. And for this, I just go to the exploration of datasets. So you can, for example, investigate the surface-based morphometry. So you see all these subjects here. So we can, for example, choose one subject, one surface-based morphometry, and select the corresponding DTI. So I want to give you an impression how easily you can work with multimodal data sets to explore results and to generate hypotheses. So we have DTI and surface-based morphometry combined. In the tabular listing, you see the surface-based measures, but also the degree, as I showed you before. We can then select a subnetwork, and one subnetwork which is known to be altered in many diseases, including Alzheimer's disease, is the default mode network. So NICARA now searches the ontologies, which regions of interest belong to the default mode network according to literature. And then we have filtered here to 36 regions of interest, and we want to see the 3D lattice graph. And this is the result now. So you see all these regions of interest, 36, which belong to the anatomical or functional subnetwork of the default mode network. And what becomes very obvious is that you have strong connectivity in these clusters of ROIs, the frontal and parietal hemispheres here, but there is very weak long-range connectivity between these clusters. For example, if we compare this to a healthy matched control, cognitive normal control, you see much stronger connectivity here between these regions of interest. So that shows you how you can further increase prediction quality of your imaging trials, imaging studies by including DTI tractography. So the hypothesis to derive from this observation is that long-range connectivity is probably another biomarker in addition to other network measures that you can derive with NICARA and assess with NICARA to further really increase the prediction quality. With this, I would like to wrap up, going back to the PowerPoint presentation. So to review this finding, so we have voxel-based morphometry with only a very few regions of interest compared to surface-based morphometry with many more regions of interest that were significant in individual testing. So after multiple testing correction, 0 ROIs remained significant, but 25 out of 360 or out of 100 regions of interest remained significant after multiple testing corrections in surface or for surface-based morphometry. So you can really increase the power of your morphometry studies by including surface-based morphometry. And this is remarkable because we had such a small study size of just 25 against 25 subjects, and this is a reasonable number still for early studies, clinical trials, exploratory trials, when you start drug research. And more important even is that you can increase prediction quality, because just for the voxel-based morphometry and volume comparisons here, Alzheimer's disease versus cognitive normal, you see that you can hardly discriminate these two groups. But if you include surface-based morphometry, you see how these two groups become really different from each other. And this certainly may increase your prediction quality. So to summarize the results, so NICARA delivers automated processing of both conventional MRI and diffusion image data. All features can be extracted from CMRI and DWI measurements, including surface-based morphometry, including DTI scalars and also DTI full brain tractography. I hope I could convince you that surface-based morphometry can tremendously enhance the outcome of your morphometry data compared to voxel-based morphometry alone. And this is all software-based, so there is no further imaging required. You can also reassess your data using this latest software technique and increase the result of already existing studies too. So here, and this is a future perspective, what else you can do, surface-based morphometry and tractography are fully mapped onto each other. So you can use, if you have DTI data available, use this as a further imaging quality to enhance prediction quality. We can do cross-sectional as well as longitudinal assessments in NICARA, and longitudinal assessments are really the gold standard for doing clinical trials to compare baseline assessments to after treatment assessments. So we believe that your results and decisions will be better with NICARA because you can develop more sensitive biomarkers for neurodegeneration due to higher precision of feature extraction. And most important for you is with higher precision, it means that you can derive conclusions from even smaller clinical trials. Especially if you start trials and plan your studies, you don't need to take into account hundreds of subjects. If you have a much smaller number, you can also see if there's something in your data. And that means you're able to generate more predictive value at much lower costs. And at the end, last but not least, you have all the relevant data of your clinical trial in one software solution, from the raw imaging up to the statistical assessments. And since NICARA can be installed as a distributed system with a client-server architecture, all stakeholders of your clinical trial can remotely access the same tool. Now we are ready to demonstrate NICARA's superior precision for your data and show you what it offers to you compared to traditional approaches. We can assess or reassess your clinical trial data in an economic and low-risk proof of concept project, if you like, but also, first of all, give you a demo access to the system, or of course, give you free consultation. With this, I'm at the end of this nice short presentation. I hope you enjoyed it and have some questions. So please, I'm happy to take your questions. Thank you very much for listening.
Yeah, also, thank you very much from my side, Markus. It was very interesting. And yeah, as you already mentioned, so now we have a lot of time left for our Q&A session. So I saw that there are already some questions arrived. Again, please use the live chat to ask your questions. I saw that there is also some people try to raise their hands. Maybe I would prefer that you type the questions in the chat. So Markus, first, there is a question. It's already answered, but I think it's interesting for the whole audience. The question was, can you please explain a little bit more in detail how you correct for multiple testing and which correction methods you are using? And yeah, it was answered by the questioner himself. He mentioned the Benjamini Hochberg paper, but maybe you have a few more words on that.
Yeah, this is work that we did together with our statisticians from Discovery, our data mining company in Vienna. So we explored that Benjamini Hochberg approach for multiple testing correction is very, very suitable for neuroimaging data, and especially surface-based morphometry. The idea here is that you basically correct for the distribution here of the p-values. So if you have a distribution of significantly or peaking low value p-values or low p-values, but a relatively flat distribution of higher p-values, you can nicely make use of this Benjamini Hochberg correction, because it's more appropriate compared to Bonferroni testing and correction and so on. So we explored that this is the right approach, and we are about to, we have submitted this for publication, this approach.
Thank you, very interesting. Okay, there is another question. Markus, how can you use NICARA for clinical trials with healthy volunteers? Because so the question relates, this use case you presented was on patient versus cognitively normal, but can you also use, and how can you use NICARA in a healthy subject study?
Well, you can do longitudinal assessments in healthy subjects, of course, and see, for example, how the different age groups decline over time, and compare, for example, different groups with different age categories, for example. That's something you can do, and also first of all, compare also different studies with each other, do meta studies, and to see, okay, do you have a similar decline in different studies in different cohorts, for example. That's something you can do, I would say. This is, of course, NICARA is agnostic here. So whatever research question you have, when you have data available, you can manage these groups, as I showed you in this live demonstration, and do these comparisons. This is up to you which research questions you come up with.
Okay, so this means you can also test other hypotheses with NICARA, okay, thank you. Okay, there I have another question is appearing. So the question is the technology, so the NICARA technology, already in use?
Yeah, Biomax with its technology is partner of the Human Brain Project, and so our technology is part of the Human Brain Project and connected to eBrains. So there is where we apply this technology already together with the clinical partners.
Okay, thank you. So there's another question which appeared. So okay, a question on the automated pipeline. Which toolboxes are you using for image segmentation in the pre-processing?
So we make use of FreeServer and also Cat12 toolbox for optimal image registration and segmentation.
Okay, so yeah, I hope we could answer all your questions. So right now I don't see more questions. Yeah, so I would like to thank you all. And yeah, there's one question appeared. Okay, no, this question is already answered. So the NICARA technology, yeah, it's already in use. So I'd like to thank you all for your time, and yeah, so maybe we see each other in the future. And yeah, I'd like to thank you all for your attention, and yeah, have a nice evening, morning, day, depending on where you are on the globe. Thank you.
Thank you also from my side. Bye-bye.
Bye.